We're going to get started. There's going to be some folks joining us from the other conference room in a moment. So thanks for joining this roundtable on statistical genetics and genomics. I want to introduce briefly or allow them to briefly introduce themselves, our panelists here. So we'll start with Dylan.
Hi everyone, I'm Dylan. I'm, I believe, the newest faculty here. I joined in last fall. I'm an assistant professor, and my research is on developing statistical methods for some genomics problems, including spatial transcriptomics and related RNA sequencing, that kind of stuff.
Yeah, my name is Hui Jiang. I'm here since 2011. I'm a professor in biostat. I work on statistical genomics, bioinformatics, computational statistics with problem mostly ranging from like cancer genomic to biomarker, that kind of problems.
Hi, I'm Jean. I've been here since 2020. I work on quantitative trait genetics. I work a lot in sort of high dimensional traits, or how can we look at multiple phenotypes together. I also work in Mendelian randomization, which is sort of a kind of causal inference that you can do with genetic associations. And I work a little bit in like omics associations.
I'm Matt Zawistowski. I came here in 2005 as a graduate student. I've been here for what was just terrifying, I guess. So actually, I did my PhD here in statistical genetics with Sebastian Zolder, actually. And then I worked for a couple years at the VA here in Ann Arbor, if you're familiar with it. So I took this job and I learned about electronic health records there. This was sort of before people were doing a whole bunch with electronic health records, and certainly before a lot of the people that were already in the genetics area were working with electronic health records. So I took that job. There was no genetics in that job, but I spent all my time working on electronic health records. So how can you pull information from sort of people's sort of medical history, how it's tracked through appointments and stuff. But then I came back here because we were starting something called the Michigan Genomics Initiative, which is a biobank where patients at Michigan Medicine consent to have their electronic health records linked with their genetic information. So they give like a blood sample and they get genetic information. So I've worked on that for numerous years. Also involved in sort of studies of methylation, some collaborations with people in the epidemiology department. And sort of those of you I've worked with in the summer program, I sort of moonlight in things like spatial transcriptomics, but I leave that to the experts over here. So.
Perfect. Well, let's start with, the word biostatistics is biomedicine, biology, and statistics. So I want to start with the methodology, the statistical element of it first. So can you share some examples of how you use, adapt, create statistical methodologies to help you answer the questions that you talked about in your introductions?
Yeah. Are we
it doesn't matter. Okay. Anyone can take any order.
Sure, yeah. The way I think about it is basically, so I basically got into biostatistics after coming, my PhD was in computer science, so I also come from a computational background. So basically I view it as that we have a lot of useful tools in computer science and statistics, and rarely are you going to have a problem that exactly fits those tools. Like, we would love if we could just apply some technique we learned in class, like PCA or ordinary least squares regression, to a problem. But the most interesting thing, and the way I think about it, is from a data-driven viewpoint. We start with the application problem, and we think, what are the data-driven statistical elements that we need to model? So for example, do we have missing data, or do we have some kind of like batch effect across different samples? And how can we take these models that we've learned and, methodologically speaking, adapt them to these new contexts? And then once we define that model, there's the whole algorithmic side, which is how do we develop efficient computational statistics procedures to run these on large data sets, because we're often dealing with large data sets for these types of problems. So that's how I view the overall process, and yeah, I'll leave it up to you all.
Yeah, totally agree with what Dylan said, probably follow the same sort of a framework. But talk about like statistical methods, I mostly work on statistical computing. I mean, I actually have a similar background. I did a bachelor and master in computer science, and then my PhD was in computational mathematics before I came here. So a lot of times when I model those problems, I mentioned I do research on cancer biomarker. I guess that's answer the next question, but the idea is that you measure a lot of things about cancer patients, like all their genes, 20,000 genes, right? In the end, you want to find a few which are sort of playing a role in the particular cancer or disease they are playing, but how do you find them? Because they are very complicated. That's turned out to be a variable selection problem. And then there are many different approaches in the field developed for that. One of the class approaches is called the penalized regression. And I widely use those in my modeling. So all kinds of penalty combined with all kinds of like loss function, sometimes even with some constraints. So you end up with an optimization problem because you want to optimize that penalized likelihood. And then how to solve that kind of optimization problem efficiently because you have big data. That's the kind of research I work on. I work on a serious number of algorithms trying to optimize those kind of problems.
Do you know Matt?
Yeah, I guess it's sort of similar to Dylan and Hui's answer. I think, right, I came to biostatistics through statistical genetics. So I started working in a stat gen lab first and then realized that like all of the things that I thought were most cool about the work that I was doing were actually statistics, and I should go to graduate school and learn statistics or biostatistics, and I ended up learning biostatistics. And I think, I think along the lines of what Dylan said, something that I really enjoy is that you start with like a biology problem, right? So you're like, I want to know, something that we've been working on is like, I have all of these different phenotypes, right? I know how tall people are and like how strong they are and maybe whether they get diabetes and like on and on and on. And I think there's like some like core systems that I don't observe. And I want to be able to characterize like what genes are driving sets of traits together. How do I find that? And what the data I have is like genetic associations, and maybe I have like some molecular details. So I can tell you that problem, and now I need to turn that into a mathematical model, right? Where the answer to my problem is some parameter of that mathematical model, right? The answer to my problem is now going to be a function that takes in like the empirical distribution of all of my data and spits out a number, right? And so that's my job, is to be able to translate that biology problem into a model and then figure out how to fit the model in a way that is efficient enough for me to actually do it, which I think is very similar to what Dylan and Hui said. But that's sort of where that connection comes, is sort of knowing the biological system well enough to create a model that answers the biological question.
So I was just going to say, in terms of methods development, because people hear that for biostatistics, and I think sometimes people imagine methods development as somebody sitting in their office with the door closed and they're just like working with pencil and paper, and they're not actually interested in applications, right? Because I hear students often be like, no, I want to work on cool applications. I don't want to do methods development. So I think the way to think about it actually is the people that are doing methods developments are usually working on applications that nobody's seen before, right? It's like, here's some new kind of data, new assay, new technology that's pumping out data that nobody's ever worked on before, right? And sort of thinking about, how are we going to do this? Because the tools that we have don't exist. I'll give you an example. When I was working on my dissertation, people were like being able to sequence genomes. People could do it for a long time, but you couldn't do it cheaply for like thousands of people at a time. But it was coming on board. Everybody knew it was going to happen, right? So when I was working on my dissertation, we started thinking about, methods for sequence data. We weren't applying them quite yet because we didn't have the data. But once it became available, right, the people that were able to jump in there and start working on this, like the new set of data, were the people that had already been simulating it and thinking about some of the issues that would pop up. So, especially if your end goal is like PhD level work, when I think of methods, I think of like, okay, you're now going to start working on some problem that nobody's ever seen before, some new kind of data that nobody's ever seen before. answering that question.
Yeah, awesome. Let's move to the cool applications part of it, because that's the exciting part about genetics and genomics is you can do so much with it. So what are you currently doing, and then what are you excited to be doing in like the very near or maybe even long-term future with technologies and methods that are becoming available now?
So I'll provide like one example of this, which will also providing some more detail on the first question. So one example of a new technology that's come out in the last 5 years, which is really promising, is called spatial transcriptomics, where you can actually take a tissue, and such as brain or cancer, and traditionally we've been able to image that, whether it's for like medical imaging or research, but now what we can basically do is take many different images of the same tissue, like 20,000 different colors, and with those, each of those images represents a different gene. And you have about 20,000 different genes in your body, and this will allow us to understand like spatially what each of these genes are doing and how they're related to organization of the tissue, including how things go wrong in disease. So one example connecting to the earlier is with this new technology, pretty much what we get is that individual measurement points, because it's spatially indexed, you can actually have multiple things appearing at one location. So in statistics, we can connect this to a concept called mixture or an additive mixture. So what we have to do methodologically speaking is, let's say we're trying to create some spatial model, and we've learned in our class about linear models, generalized linear models. Those models typically assume that you have to take a single measurement per data point. But here, now we have the mixtures, so how do we adapt our model to that? That's the kind of thing we think about. And then algorithmically, like Hui mentioned, we have some algorithms for the original problem, such as iteratively reweighted least squares, Newton-Raphson. Can we adapt those same algorithms to the modifications we've made? And that's the computational statistics aspect. So I think this is one example of a technology, the spatial transcriptomics, that we can use to study the spatial organization of tissues in a much more high throughput way. And some of the applications of this are that by screening all these genes, we can find biomarkers that we can use to either, like, for example, in cancer, diagnose or detect cancer earlier, and take this from a research tool into the clinic. So I think that's where the field is heading, I believe, in the next 5 to 10 years, is taking these kinds of new technologies from a research tool to actually helping people in the healthcare setting.
Yeah, I'll talk about one specific problem we have been working on recently, and had a first draft of paper just submitted, and actually got accepted by a conference. My student is going to Minneapolis like in May to present about it. So it's a project we started a couple years ago. The idea is to find some gene, which is driving some cancer. People do that for decades. But the challenge is that, a lot of times is cancer is still, nowadays actually a common disease, but specific cancer, especially some rare type of cancer, is still rare. It's rare in the sense that it's hard for any institute to have a large sample size, right? For breast cancer, lung cancer, we have thousands of people in a particular hospital, but some rare type of cancer, actually you have hundreds, even less, right? With that small sample size, really limits your power to find what's driving them. Right? No matter what you do, you don't have the sample size there, not much you can do. So the idea we had in that project is we know that actually a lot of cancer in the end, they are driving very similar biological pathways behind them. So it's actually the same gene or same group of genes actually driving different kind of cancer in different patients, but only a subset, not all of them, right? So how do you borrow strands basically across cancer, pull them together? Say maybe we have each cancer set type is small, but when we put them together, we have tens of thousands of patients, right? Then we have a larger sample size. We have a chance to find some gene which is driving more than one cancer type. But that's sort of a problem that in statistics is pretty hard, because typically you assume either the gene is not driving nobody or driving everybody, but driving some cancer but not the rest. How do you even put them, right? If you just naively put them together, analyze them as one data set, you actually lose power because of the heterogeneity So many people in recent years start working on this data integration with sort of heterogeneity building in. We come up with a method which trying to give different data sets a weight automatically based on the effect size, right? Say there's a cancer you want to work on but has a small sample size, but if you have so many other cancers that you can pick from, you're gonna pick the one which are similar in the sense that they have sort of a structure, right? But how do you find that similarity? You have to run a lot of sort of we build a model in the end using cross-validation, trying to find the cancer which can help here, but that turned out to be a huge sort of a computational problem. We actually use the cluster, we optimize the loss, still take a month to get preliminary results, which we are summarizing. So, luckily we did find something that by joining them that we couldn't find by looking at them individually. So, that's one example we recently working on with the real application.
So I'll talk about something we've been working on for the last year-ish. So we've been working on this causal network problem where, so I guess the very simplest causal network is just two traits, right? And I want to know does trait 1 cause trait 2? And we being the whole field of statistics have thought about that problem a lot, right? Just this two trait problem. So then you can get a little bit more complicated and say, I would like to draw a causal network with lots of traits. And this connects to genetics because we're going to do it using genetic associations. And so we're going to sort of take advantage of this instrumental variable paradigm where we use our genetic instruments or genetic variants to tell us whether we think we have causal effects from one trait to another. So we've been working on this problem for a while, and we now have this method where if I think I know the network structure, I can estimate all of the effects in the network up to a couple hundred traits. So that's been working for us we sort of just have gotten this going in the last year that we can fit this network. And then there's this extra problem of I don't actually know the network structure. So I want to discover the network structure, but I can't just try all of the networks because if you start thinking how many different networks are there that connect n traits, you will find out the answer is extremely large. If n is bigger than 5, it's a lot, and it grows super exponentially. So I can't just try them all. So we've had to explore different strategies of how do I explore graph space efficiently so I find all of the best networks and I'm able to give you, we are going for a Bayesian strategy. So we're looking for posterior summaries or posterior probabilities of different network structures. So we've been working on some sort of strategies for taking graph space, which is this very discrete space, and how do I make a continuous representation of graph space so that, because I can fit things that are continuous a little bit more easily than I can fit things that are discrete. So we now have a working version of this where we can actually do a pretty good job of finding the causal network with a few dozen, maybe 100 traits. And we're hoping to push this into, I would like to actually do this with thousands, right? Because I've got proteins and I have 1,500 proteins, right? I'd love to be able to just draw the causal network between all of these 1,500 proteins. We currently can't do that with our, even with our much more efficient strategy where we don't have to look at all of the graphs, we still can't quite handle that scale. So we're sort of trying to push that scale up so that we can start to integrate metabolome and proteome and maybe gene expression. And sort of the ultimate goal is you have this big network, and you might even be able to do stuff propagate effects through and say, what happens if I perturb the level of this protein, what are all the things that I think are maybe going to happen, right? And that might help me do something predict side effects for a drug, or predict new drug targets for a disease, or test hypotheses about how a disease mechanism might be working. So that's our the pie in the sky extension from where we are, which is we can sort of do this for 40 traits right now.
Thousands.
Yeah, why not?
Why
Once you
not?
can do 40, let's do thousands.
I thought you were going to say hundreds, and then you go with thousands.
Yeah.
All right. The cool project I worked on recently, because it was so different than anything else I've ever done, was a collaboration with some people I have at Vanderbilt, and they were a clinical group. And they were interested in looking at clinical decision making. And so they were looking at this drug. I won't pretend to remember what the name was or what exactly it does, but it was used to treat an autoimmune disorder. And one of the known side effects of this drug was that it could lead to big drops in white blood cell counts, right? And if your white blood cell counts drop too much, they have no choice. They say, we got to take you off of this. And there's a relatively arbitrary decision rule on if somebody's white blood cell drops below this, we're going to pull them off this drug, right? So then they lose the potential therapeutic benefits of being on that drug. And so their hypothesis was that, this is not really evidence-based. We don't know why this is happening. We're just sort of think this is a good cut point to use. And so what we actually showed was, so we used this biobank data. If you wanted to do an actual experiment of this, it would be pretty difficult to do, right? You'd have to sort of change people's, they're being treated for disease. Maybe you have to randomize that, like this drug works, right? So the idea was, well, let's use historical data that's already, people have already been prescribed this drug. We've seen what's happened in the past. Let's pull that from their electronic health records and almost do like an observational experiment, right? And so what we did was basically looked who was prescribed this drug. They had white blood cell counts measured, as part of their course of treatment, and so we were able to track that. And then, because they were genotyped, we had genetic information on them, we were able to predict what their white blood cell count should be, right? So if you imagine, everybody's sort of born with a handful of genetic variants that sort of determine where your white blood cells will be roughly, right? Your sort of inborn value, and then, different environmental exposures will change that. But what we were able to show in this was that these clinical decisions were being based more on what somebody's initial genetic risk was for white blood cell counts rather than their actual response to the drug. So what I mean by that was, people who started on the lower end of white blood cell counts and were completely healthy, right above that threshold, they would start taking the drug and their white blood cells would drop a little bit below that threshold and they get pulled off the drug right away, right? But really, this wasn't a problem for them. Their natural biology was to be sort of on the low end. And then there were people who were, had very high white blood cell counts, naturally. They would plummet, but stay just above the threshold, right? And so, this is a problem when you have that much of a change, but it didn't cross this arbitrary threshold. So it was pretty cool. And so, the point of the paper, the clinical folks, they were like, this is what we need. And so, to publish this, it got published, and the hope is that it's going to lead to changes in how this kind of drug is administered and, how side effects are viewed.
So did you come up with a new rule?
Well, I'm hoping they do.
Okay.
Yeah, so I think that the advice was we need to consider what somebody's baseline white blood cell counts are. It's kind of surprising. In some of these cases, they don't even start measuring white blood cell counts until after they start taking the drugs. So they're not even looking at changes like that. And so again, this is one of these places where I think the electronic health records, people were just collecting this health data, and people have found ways to use it to do these kind of sort of natural experiments.
All right. Well, thank you all for your discussion on the Statistical Genetics and Genomics. Students, if you want to continue having conversations with these faculty members, they are going to be going back to the lunchroom, and you're welcome to have further conversations with them and ask questions there. So we are going to do cancer biostatistics next in this room, so if you want to stick around for that, you're welcome to. Otherwise, thank you so much.